Hi everyone, I'm Jaya Vishwanathan, and you've found the No Brainer podcast where I discuss all things neuroscience in delightful conversations with various experts. I hope you enjoy today's episode.
I want to add, and I really want to get your opinion on it because, I'm talking, this is coming from my perspective, which is not as an immunologist, but one thing I've always felt is that, we evolved as a species to get past the reproductive age. And so humans are living much longer than we did when we first evolved to have all these complex cognitive abilities. And so to me, it always seemed like perhaps, and correct me if I'm wrong, but perhaps because the immune system has such a big job to do to keep us healthy and safe, it's almost they're overactive all through like adolescence and midlife. And so they kind of stay in that overactive mode as we go into aging, which is why there's more and more of these autoimmune diseases that are happening. But that's maybe a little bit more philosophical because I don't know if there's been proof. I really genuinely don't know if there's been proof about that. And so if people are just tuning in now, I am talking with the amazing Dr. Malú Gámez Tansey about the complex biology of the intersection of the immune system and the central and the nervous system, not just the central, but also the periphery, the blood-brain barrier, and the crosstalk. And to also add another level of complexity to this conversation, as if it wasn't complex enough, I would like to hear from you about something I know I'm interested in, you're interested in as well, which is like gender differences in the brain and immunology. We know that a lot of these neurodegenerative disorders, especially Alzheimer's, are sexually dimorphic, and that the disease etiology, the disease biology is fundamentally different in male brains versus female brains. And just so any insight about that would, I think, really be interesting to our audience.
Yeah, so you mentioned a couple of things which, in fact, have good foundation on biology, and that is that as we live too long, right, maybe our immune system is exhausted. And there is a term in immunology called immune exhaustion, and we've heard about it in the context of cancer because T cells become exhausted. They start expressing PD-1 and other markers of exhaustion. And immune exhaustion is when they just basically cannot do their job anymore. So they lose immunocompetence. Some of it may be related to senescence, but not necessarily. And one of the interesting things that Rebecca Wallings in my lab, who I'm hoping is on her way to independence, thanks to a Parkinson's Foundation Launch Award, she'll be coming to Indianapolis with me. And basically, her work found that the LRRK2 mutations, which are the most important ones for familial Parkinson's, they are interesting in the sense that when you are young and in a young mouse, those LRRK2 mutations make your immune cells work overtime, like really well. Your lysosomes are great. Your pathogen control is great. Your antigen presentation is great. They are like working overtime. And we think that that may be the reason why some of these mutations persisted in the human population, because they give you an immunological advantage when you're young. But if you live too long, then they're exhausted. And Becky has data suggesting that these mutations confer a type of immune exhaustion. There is a difference between males and females, which you'll be interested in hearing. And when we look in the humans, we see the same phenotypes, that individuals with these LRRK2 mutations, their peripheral blood mononuclear cells, or PBMCs that you get from the blood, have features of immune exhaustion. We've become very interested in understanding, is that a mechanism that also applies to sporadic non-genetic disease? And the exciting thing is that Becky has been able to also discover that chronic inflammation can trigger immune exhaustion of your peripheral immune cells, which basically says that maybe that's one mechanism by which you contribute to these age-acquired proteinopathies because the vacuum cleaners no longer are doing their job. They are exhausted.
Yeah.
They're out of juice.
Yeah.
And so that's a really interesting thing that you said, we've lived too long. They do become exhausted. And so that is an area, that we should all watch for, because we and others will be working in this space. And the advantage there, if there is such a thing as immune exhaustion, it's then possible that we might be able to leverage some of the checkpoint inhibitors that the cancer field has been using to see if we can parse out the kinds of patients in a precision medicine type of approach that might be good candidates for those, right? So we take their blood, we look at their PBMCs. Are they showing those phenotypes? This drug might be good for you. So in terms of personalized medicine, that's what we want to do. Not everybody's inflamed. Not everybody with Parkinson's and Alzheimer's has underlying inflammation. But for the 60 or 70% that we have, measured do have an underlying inflammation, and it's not like raging inflammation, it's just low dose chronic inflammation. For those people, the hypothesis is that the underlying inflammation is driving their disease progression. And so even if you already have the disease, we're hoping that we can arrest it by targeting the chronic inflammation, right? And it may be through diet and exercise. It may also be through better living through chemistry and targeting specific pathways like soluble TNF and LRP3, others that are likely driving the disease. That's the hypothesis that has been tested in the preclinical models of, animals. But ultimately, you have to take your tool, your drug, and test the hypothesis in the clinic, in humans, and see whether or not it translates. And so we're very excited about that, because it will give us an opportunity to survey peripheral immune cells, which are easily gotten to, right, compared to, say, CSF. Find out whether there's dysfunction. And the way we do this is that we plate them in a dish, and then we challenge them with various things, immunogens, pathogens, and we basically ask, are they playing with a full deck, right? Or are they dysfunctional? And then we throw a drug in there, and it either fixes them or it doesn't. And if it does, those are the people you enroll in your clinical trial. So it's a way to stratify patients that have underlying innate immune dysfunction or adaptive immune dysfunction, which can then be taken into a trial with specific drugs that are likely to cure their condition, much like the cancer field has done it, right? So we're not the first to think about it. We're just borrowing a page out of their playbook.
No, I mean, you're still the first to think about it in this space. And, I think there's just so much, there has been historically too, just so much frustration with neurodegenerative disorders, because the prognosis is so bad, and it really, so much of what we consider as being human comes from the stories that we tell ourselves about who we are.
Of course.
Even though, in some respects that might just be like a byproduct of consciousness or higher network functioning. But even so, that's so fundamental to who we are. And the problem is that, unlike something like diabetes or even heart disease, which is devastating, something like dementia and Alzheimer's, it really messes with our conception of what it means to be human. And it's just
100%.
so heartbreaking to see someone you love slip away over 10 years, right? And so I think it's very, very important to start thinking outside the brain and outside the box, as you said. And I love that. So coming full circle back to what you started with. And so what advice would you give to people who might be interested in neuroimmunology or even just careers such as yours. And now I'm going to come to that question about your pivots from, going from academia to industry, and if you have any kind of advice for what leans people towards success in one versus the other versus what, that
Yeah,
would be, I think, very useful.
sure. So, one of the things that I mentioned before is that you've got to do what you're passionate about, right? What
Mhm.
really, trips your trigger. You just got to follow that, and you've got to,
Yeah.
play detective in a place where you're really interested, right?
Yeah.
In terms of a student, I would say it's not important exactly what project you work on. You just have to
Mhm.
learn how to ask good questions and how to design good
Okay.
experiments and interpret your data. Um,
Okay.
so look for the environment that will allow you to do that. Um, don't worry about the exact project. Their projects come and go, and really you're being taught the tools for coming up with good hypotheses and testing them.
Mm.
You can publish a few papers and you're in and out, right? Um, it's nice if you publish more too, but really the job of a mentor with a graduate student is teach them how to ask good questions, how to design
Mm.
good experiments with the right controls, how to interpret their data, and then send them off to greener pastures, right? They don't make
Right.
that much, so they can't be poor for that long. Um,
Yeah.
and then as a postdoc, you need to look for the environment that's going to launch your career into whatever,
Right.
right? And for that, it matters more, what have you done for me lately? How many papers, how many assays, how many, you know, different methods you've developed. It's a
Yep.
deliverable, right? Because
Yep.
people who want to hire you, whether it's academia or pharma or policy, they want to know that you can take risk. They want to
Mhm.
know that you can find your own money if you had to. And most importantly, they want to know that you can start and finish something. So the
Right.
number of papers is important because it's the currency, but mostly it tells me that this person is a finisher. And that's
Yeah.
really important. And you can start a million things, but if you don't finish them, it's no good, right? So I think as a person interested in science or immunology, I certainly wasn't at the beginning, and I certainly
Mhm.
never thought that I would come back to academia. I did it because I missed the mentoring. I really missed,
Mm.
you know, working with students and seeing their data and saying, well, what do you think, right? And
Mhm.
a lot of us got started as technicians, and
Mm.
technicians get told what to do, right?
Right. Right.
In my lab, I challenge them, and many of them have their own projects now. But generally, it's like stain this, whatever. Don't ask any questions. Follow the
Yeah.
protocol, right? And
Yeah.
then when those technicians become graduate students, I used to have some that would come and say, "Okay, what do you want me to do next?" And I'm like, "I don't know. This is your project. So what do you
Yeah.
want to do next?" And so you try to give people enough room to be creative. You try
Mhm.
to not let them stray too far, off the main path, but you got to give them room to just kind of experiment with, the kinds of stuff they're going to want to do, um, and
Yeah.
also how to deal with failure and rejection, because most science doesn't work, right? Most experiments
Yes.
don't work. And to get
Yes.
comfortable with publishing null results, right? Get comfortable with, hey, it was not as exciting as I thought it would be, but it's an important part of knowledge. And you and I were
Yeah.
at that workshop, which is so important and
Yeah.
critical that people publish their null results,
Yeah.
because that knowledge is lost if they don't. And so
Absolutely.
I think that's an important thing, to be happy with what you're doing, to want to jump out of bed and get in the lab, check on your cells, check on your animals. Um,
Yeah.
if you don't feel that passion, that's the most important ingredient, right? I don't really
Yeah.
care how smart you are, how hard you work, how lucky you are. It's how passionate are you about this line of work and asking these questions? Because most
Right.
people who are good detectives, you can get them interested in anything, right? I
Right.
didn't do neuroscience as a graduate student. I did smooth muscle with an
Mm.
amazing mentor who taught us how to ask good questions. And as a
Right.
postdoc, I did, dopaminergic neuron neurotrophic factors. And then as a,
Yeah.
faculty, we started with inflammation. Now we're working in the gut-brain axis. Like, who knew? Who knew?
Right.
And
Right.
honestly, you are data driven. And in my case, I'm driven by the aspirations and ambitions of the people that I'm training, because
Mm-hmm.
doing that is going to help them do what they love. And
Right.
so, and so, depending on what stage you're at, you're going to
Yeah.
need a different mentor. You're going to need a different environment. Um, don't feel bad, right? Look for mentors outside your institution. Look for mentors
Right.
across the world. There's a wonderful mentoring platform that was developed by Robin Chapman called the Thrive Mosaic. And if you
Okay.
Google it, Thrive Mosaic, you'll get the paper she wrote. Her grandmother was a civil rights activist, and she's
Mm-hmm.
African American. And her grandmother used to say, never pass up an opportunity to overcome an obstacle. And what she devised, what Robin devised, is a mentoring scheme with about 5 buckets. And the
Okay.
bucket of mentoring are associate, which is basically someone at your level, right? Someone
Okay.
that you can go to and say, hey, what do you think about this? And they're
Mm-hmm.
like, oh, no, 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 don't do that. Don't commit to that. Say no. So
Right.
it's our no tree, right? In academia, a lot of women have no trees, right? Tell me to say no. And so that's an
Yes.
associate. You also have a
Okay.
sponsor. You have a
Yep.
connector who can connect you to various people.
Okay.
You have a
Okay.
targeted coach who can maybe teach you about budgets or how to give a chalk talk or you name it, right?
Yeah.
You have a mentor like an advisor. And
Right.
what is so nice about this scheme is that it's less daunting than just asking someone, will you be my mentor? Right? Because most people kind
Right.
of go, oh my God, what does that mean? And they run for the hills. But if they
Hmm.
know that all you need is for them to connect you to so and so, or for them to teach
Right.
you how to do budgets, right? It's a deliverable. It's a much easier ask. And so this
Absolutely.
framework was developed for marginalized groups in STEM. I think it's applicable for anybody. And so when you
Right.
go to a conference, when you go to a reception, instead of being concerned about, who am I going to meet and what am I going to say, as you talk to people, you can start thinking about, how could this person fit into this mentoring scheme? Could they connect me to someone?
Hmm.
Could they teach me something? And you start to put this Rolodex in your brain. Right now, I'm really dating myself with the Rolodex analogy, but you start to file them under different buckets, right? File
Right.
this one under connector, file this one under, you know, targeted coach. Um,
Yeah.
and that way, you really build this village of mentors. And I think that is what really has helped me and has helped a lot of
Mm.
my trainees, right? They say to me, hey, could I reach out to so and so? By all means, right?
Yes.
We're not the experts on everything. Please don't
Right.
reinvent the wheel. Um, get an adult in the room and have them teach us stuff. So I think
Right.
you have to have the humility to park the ego at the door and say, I don't know anything about that. Like,
Yeah.
who can help us, right? And that's
Right.
how I think we've developed a lot of our collaborations, a lot of our new learnings, and
Yeah.
hopefully some impact, across.
Yeah. No, thank you for that. I will definitely put like the Thrive Mosaic as well as your Twitter. You're very active on Twitter, and I follow you myself. And
Great.
any other contact details for you that you would like to share in the show notes, because I think that's just such valuable information for people to have about, maybe networking and mentoring tips if you're new to a field and trying to get your foot in the door, which can be really challenging, as we know. And
Absolutely.
I do want to say, I think that you are timeless because your ideas are just all encompassing, and I think every time I speak with you, and I know, and I we have mutual friends and colleagues, and like every time any of us talk to you, or you're at this conference or that conference, we all learn from what you have to say. And just a funny anecdote is that we met because I work as a contractor at the National Institute on Aging. I work on this platform called AllSped, and we did a survey of the literature in the Alzheimer's preclinical or animal model studies testing candidate therapeutics, and about 3 to 5% of these published studies have done things like, very basic things of research studies, like doing power calculations to adequately have statistical significance, or just doing the one part of the study with like male mice, and then doing a completely different part of the study with female mice, and then drawing a conclusion that, oh my God, this drug works, or things like that.
Yeah.
And we ended up commiserating on how terrible the landscape can be. But that's just a little aside for those of you who might be interested.
Yeah.
So I'd like to maybe chat a little bit about your future projects that we can look forward to, and I would love to hear about, and I'm sure the audience would as well, about the book that you're working on, which I know is coming out, and I cannot wait to read it. So yeah.
Yeah, so future projects. Again, I think my excitement about moving to the Stark in Indianapolis is that it's an environment where I think the people whose careers I'm still trying to develop into independence, where they will thrive. When we visited, I took them, they loved it, they gave short talks, they got feedback, and they could really see themselves doing a good job with their own individual careers. So that was really important to me. And the environment always, and your colleagues are everything. And so we've been really wanting that kind of environment. And the University of Florida has been amazing in helping us, build this next stage of our work after Emory. They've provided so many resources, and we've been very successful. So without this stint, we probably wouldn't be, as attractive or as sought after by IU if we hadn't been here. So I'm extremely grateful to the Fixell family for their support on the endowment, and we hopefully gave them, a return on their investment. The future will look a lot like the paths that people like Becky Wallings, Andrea Merchant, Kelly Maniz, and Valerie Doors, who are the four amazing women that are coming with me, will decide. And they're going to consist of the role of immune exhaustion in, in age-acquired neurodegenerative diseases, the role of the mutations that we know confer higher risk for various neurodegenerative diseases in immune exhaustion and immune senescence. We're going to do some epigenetic studies to figure out, how is it that, the epigenetic regulation of peripheral immune cells is a way to kind of imprint some of the things that the environment does to you, exposures, diet, and things like that, that is not encoded in your genes. We're going to be looking into the role of the gut-brain axis in autoimmunity. Andrea Merchant is very, very talented research faculty, assistant professor at IU, who came from UVA, from an amazing lab, an amazing neuroimmunology environment there in Alvin Gautier's lab, and she's developing that line of investigation. Kelly Maniz has been doing a lot of work with synuclein and the role of synuclein in immunity and inducing inflammation. So she'll be doing more of that. And then Valerie Jors has been working on cannabinoid signaling, the CB2 receptor, and how it could modulate removal of tau, removal of synuclein, removal of amyloid. And that's a really hot area, right? Because everybody's like, well, marijuana has so many benefits, but you don't want the psychotropic effects. So how can we leverage the ligands for the CB2 receptor, CB1 receptor, cannabinoid, 1 or 2 receptors to come up with a pharmacological way to derive the benefits. So that's Valerie Dors's platform. And I think we're going to be also definitely wanting collaborations with the immuno-oncologists at IU. Very excited about that.
Right.
Director of Neuroimmunology Research, that's going to be my job, to bridge those two groups and have us talk more together. And we'll also have, lots more interactions with the Lilly people who are developing these drugs. And so Mike Hutton and others, we've had some discussions. So I think it would be very fruitful, I'm hoping. And then in terms of, where else would we go? We'll see where the data take us, right? We're going to be data driven, and we will look for collaborators that want to learn from us and want to teach us a few tricks. My book. So it's funny that you brought it up, because one of the other things I wanted to mention in terms of mentoring and, how to move through academia or really any science workplace is this idea that, when you start to introduce yourself, you say, well, my path has been unconventional, right? Well, my path has been unconventional.
Right.
And I come to the conclusion that everybody's path is unconventional. There is no straight line, right? You may lay out the best plans, but life throws curveballs at you, and you end up in places where you never really thought you would or intended to, right? And so I consider myself not a destination-driven person, but more about the journey. And so for me, I started out at Stanford. We moved to where my fiance at the time got into med school in Texas. He got into one med school, I got into 5, and there were 6 in Texas, and we did not get into the same one, right? So I thought, you know what? This is the second time you're applying. Let's just go.
Yeah.
And I'll work for a year. And I worked as a researcher, and I fell in love with research. Like, there's no way I'm going to go take care of sick people. I'm going to do research, right? And so I applied to grad school, got my PhD, and I never regretted it, never looked back. And then he was an MD PhD, so he matched at WashU, right? And so it's like, well, we're going to St. Louis. I'm like, okay, great. I had no idea who was there, what was there. So I started looking for postdocs there, right? And I found incredible labs, lots of options. It was hard to pick because I could get excited about almost anything that's worth investigating.
Right.
So I went. And so then from there, his thought was, okay, well, this gig is over. Now I want to go to UCLA for a rehab fellowship at UCLA. Okay. And I said, okay, well, I'm going to try biotech. And so we went there, and it turns out the first company I ended up with was a disaster because I didn't ask all the right questions, and it's closed now. I'm not sure whatever came of it, but they were not really doing good science, and there was no good scientific leadership. That's all I will tell you.
Okay.
And so ask questions, make sure, the company is as good as the scientific leadership. The institution is as good as the scientific leadership. So look to the scientific leadership and ask a lot of questions. And so I left and went to Xencor, which is the one where we developed the TNF inhibitors. That was the best, and still continues to be the best.
Right.
I feel like I've seen, a tale of two cities, the best and the worst in the span of 2 years. Right. And so I learned a lot. I learned that there's a lot of rejection. I learned that, for people of color and women, there's still a lot of obstacles, right?
Yeah.
You see the memes with like all the, hand grenades and obstacles that it's all true. It's
Absolutely.
all true. Absolutely. And I never noticed it until I became a faculty member. I didn't really feel discrimination in high school, didn't feel it at Stanford. It was until I became a faculty member where I could see that it was tough to have a seat at the table.
Yes.
I could see that, there were a lot of manals, and that the administration was all, one race, one gender, and, one sex, and there was nothing else.
Yep.
So 20 years later, what I can tell you and what I tell trainees and what's going to be in my book is that it doesn't get any easier.
Yeah.
It gets harder as you keep climbing the ladder of, responsibility and success.
Yeah.
But you get better at it. You get more resilient. You put on the Teflon coat, you build networks.
Yep.
You learn how to not give a darn about someone who's bent out of shape at you, you know. And so I think it's important to walk in with your eyes wide open.
Yeah.
Right? It doesn't get any easier when you become faculty or when you get your first R1, because guess what? But you get a second one, and it doesn't matter that you've graduated one student, you need to graduate of several, right?
Yeah.
There's always, it's always a moving target, but what is really important is be happy with where you are now. And it sounds like cliche and trite, but listen, there are no guarantees that you're going to live past next week, right? So what you're doing today and how you're interacting with the people and motivating them to go ask that next question or put that rejection letter for your paper away and use it to improve it. And let's find another journal, right? Let's find a place where your readership is. Don't worry about the impact factor so much.
Right.
It's where are the readership? Where's the readership? Because it's people that are going to read your paper, that are going to download it, and they're going to cite it. Nothing against Science, Nature, or Cell, but they publish a lot of different things. The chances that someone's cruising the science, the table of contents of Science and goes, oh, this one, very little, right? But if you go to a specialized journal, some of the journals for the societies and the neuro journals and the neurodegeneration journals, that's where your readership is, right?
Yeah.
And so think about that. Of course, impact factor is going to play into, consideration with tenure and promotion, but it's really overrated. It really, you need to look for your audience and where your science is going to make the biggest impact. If you look over my publication record, we don't publish just in Cell Science and Nature. In fact, I've published in Science once, the dominant negative TNFs, and never again, right? We published a few in Nature Neuroscience, Nature Medicine, Nature this, and now Nature Parkinson's Disease, which is where I'm an editor-in-chief. We're very proud of that journal, and we don't get any special treatment, so that's where our readership is for a lot of our Parkinson's work. So I think it's important to put things into perspective and to know that this path that you're going to follow is definitely not a straight line, that it's windy, that sometimes you have to reinvent yourself. And, if you get bored with what you're working on, we'll work on something else, right? It will cost you a little bit. It will slow you down a little bit. But ultimately, you don't want to work on stuff that you're bored with.
Yeah. And you learn from it.
Absolutely. My postdoc mentor, Jean, used to say, well, it's not great to get scooped, but, you could work on something that nobody else is working on, and you never get scooped. But then you got to think about, how important is it if nobody else is working on it?
That's very fair. Yeah. Perspective. Yeah.
The other thing is, think about where the hockey puck is going to be.
Right.
The field is here now. If we solve this problem that so many people are working on now, what's going to be the next problem that's going to have to be solved?
Right.
And so for people who are starting out their labs and need a competitive edge, don't flock to where everybody else is. Think about one step ahead of what might be needed, right? So that you get a little bit of a head start. Think out of the box, take a few risks, but try to focus, right?
Yeah.
That was something I could never do. My graduate mentor used to say, focus. I'm like, and so we ended up doing inflammation and this, that, and whatever.
Yeah.
And to be honest, it has taught us a lot. The differences, the similarities, and from a funding perspective, it's really good to diversify your funding portfolio, because you're not just relying on NINDS. You can go to NIA, you can go to Michael J. Fox, you can go to Parkinson's Foundation, you can go to ALS or FTD. And so diversifying your areas will help you diversify your funding portfolio.
Right. It sounds like your book is going to be something of a toolkit for people to navigate their careers, and I hope so we'll have, whenever it comes out, let me know, and I will update the show notes to reflect it. And I hope you all get a copy, because I will definitely get mine and bug you to sign it at some point, Malú. But I can't
100%.
wait to read it, and
Thanks.
I think it's going
Thank
to
you.
be such a valuable resource for people at all stages of their careers. And in science and academia, but also just anyone who is maybe not as happy in what they're trying to do for work and trying to think about what brings them joy. And I think your book will have, from based on your lived experience and your wisdom. Thank you so much for sharing all of your scientific and life insights with us today. My heart is full and my brain is bursting with ideas, which is what happens after every conversation with you, Malú. And so thank you again so much. It's been a real pleasure. We're going to have you back because I'm sure there's going to be a lot of interest and follow-up questions. About more neuroimmune and gender differences and evolution and all of the various topics that we've covered today in life. So thank you again. It's been a real pleasure.
Appreciate the time, and I look forward to coming back.
Perfect.
Hey everyone, hope you enjoyed this episode of the No Brainer podcast, and see you at the next one.
Discover the magical world of senses with Baby Senses, a sensory neuroscience primer for all ages. Journey through the wonder of sensory systems, from traditional human senses to extraordinary animal abilities, in this wonderful award-winning children's book by Dr. Jaya Vishwanathan. It brings to life a world hidden from the naked eye by blending neuroart with neuroscience to reveal nature's complexities and marvels. Baby Senses is an enchanting exploration of how we perceive our world. With a helpful glossary at the end, this book is perfect for young minds and curious adults. Dive into this beautifully illustrated masterpiece and awaken your senses to the beauty and diversity of life.